we are going to share the results. Then we shall briefly look at RStudio, and also the assignment that was given yesterday is very important. So we shall be going through the assignment together so that we can see if we have any challenges with installation of the software that we need to use. So I'm sharing my screen now. Okay, so yesterday we requested each and every one of us to participate in the pre-survey, and we have some results here. Again, we are sharing the results so that we can have an idea of the responses that we have and any information that will guide us to conduct the training better. So the first thing we wanted to know is the role of the participants in their institution. Okay, so we have quite a number of students, over 50%. We have researchers, we have data managers, we have M&E professionals, project coordinators, we have ICT staff, ethics and governance officers, and software developers. Now, we also wanted to know the tools that are currently being used, especially because we are going to use them. We have Excel. We have quite a number of people using Excel. The percentage is quite large. We have those that use RStudio, RShiny. There are quite many as well. And then we have very small percentages using Blast for our bioinformatics. A very small percentage using the online tools like the NCBI databases. We also have few people, okay, using genotyping by sequencing tools, our bioconductor packages. Some are not using any, others are using Galaxy platform. So generally, there are quite very few people that are using bioinformatics tools for their day-to-day work. So we will be focusing a lot on them, and we hope that by the end of this training, you have gained skills in using the bioinformatics tools that we are going to introduce you to. Okay. We also wanted to know, in particular, familiarity with Excel. So quite a number of us are familiar with Excel. Quite a number are familiar with Excel. And then when we go to RStudio, we also have quite a number that's familiar with R and RStudio. We also looked at familiarity with bioconductor packages, and we have quite a number that is not very familiar or not familiar with bioconductor packages, and we shall be using a lot of the bioconductor packages during the training. RShiny, we have also quite a number that is not familiar with RShiny. We have quite a number that is not familiar with Blast, okay? According to the survey, we also have quite a number that's not very familiar or not familiar with Galaxy. Also familiarity with MGRust, we have quite a number that is not familiar. So generally what we are seeing is that most of us are not yet familiar with the tools for bioinformatics. So we are hoping that by the end of this training, you will be familiar with these. Okay, so we also wanted to know which of the bioinformatics tools and methods you are most interested in. Okay, so we have a number that is interested in biological databases. Many are interested in understanding the visualization methods. That includes the heat maps, the PCAs, the Manhattan plots. We have those that are interested in understanding sequence analysis, data quality control, population genetics, central dogma, genomics, and many other things that we are interested in. So we can see that many of the topics that we are going through already are the things that people are interested in understanding. Okay, so we also wanted to know your confidence level in using biological databases, and many of us are not yet confident in using these biological databases, but we are glad to say that during the webinar, we should be able to cover that and raise the confidence levels. Likewise, the confidence levels in sequence alignment is also not very high. We can see very few people that are confident and majority are not confident. Working with bioconductor packages through RStudio, we can see also quite number that is not confident and very few are confident. Confidence levels in metagenomics, we still have quite a number that is not confident. Now all this is expected. And in this training, we aim at going through and covering these topics and making sure that we are familiar with these. Confidence levels in transcriptomics. Again, quite a number is not confident. In genomics, we see the same pattern in the responses. Confidence level in population genetics is the same. Quite a number are not confident. In data visualization, especially for bioinformatics, also we have quite a number that is not confident. And okay, the other thing that we wanted to know is what skills do you hope to gain from the webinar? And so we have a majority are expecting to gain general bioinformatics knowledge and confidence. There are those that want to understand tools and methods for bioinformatics, and then those that are seeking to understand genomics, transcriptomics, and metagenomics applications. And then there are those that also want to clearly understand data analysis and visualization. And then we also wanted to know the challenges faced in analyzing biological data. Okay, so majority noted knowledge and skill gaps. There are those who also noted tools, software, and coding challenges. There are those who have no experience at all. And then quite a few noted data access and availability as a challenge. Yeah, so we have majority really identifying knowledge and skills gaps within the institutions. So in this webinar, we'll be focusing on addressing this knowledge gap so that we can be able to be more comfortable with bioinformatics and the tools that are used in bioinformatics. Availability of software, especially RStudio and bioconductor packages. We want to know if you have RStudio and the bioconductor packages. So quite a majority, 49.1%, said they do have. Okay, quite a number is planning to install. So we will do the installation, especially for the packages together, so that we can be guided on the packages that are relevant for the bioinformatics training and largely for the bioinformatics work as well. So those are the results that we do have from the pre-survey. So it gives us an understanding of where we are as we train and learn together in this webinar series. The other thing that I want to take us through again is the Google Drive. The Google Drive. So we have updated the Google Drive folder that contains the material that we're going to use for our training. So it includes material that was used yesterday. and material that we are going to use today and also tomorrow. So let's pay attention. We are going to download again the files from here, and we will be guided on how to open, especially the zip folders that we have, and also to make sure that we put all of them inside the bioinformatics folder that we created in R. We will go to RStudio and see that we have created these. Okay, so we have these files here. When you open your Google Drive, you can sort these, okay? So that you have the ones that have been updated appearing on top. So we are going to download the silver file. This one, the second one. We are also going to download the sequences, the RDS folder. The metagenomics packages is something that we downloaded yesterday, but we are going to run that installation today, and we shall find out those that succeeded and those that might be having challenges in running the code within this particular file here. Then you will also download the metagenomics activities for those who are running the Windows platform. You will use the one for Windows. For those who are running the Mac operating system, you will use the one for Mac. So we are downloading 1, 2, 3, 4, 5, 6 files. You're going to download them. You can highlight all of them.
Please, also metadata file in Excel.
Oh, and the last one is also the metadata file. You can also download it. So you can download one by one, or you can highlight all of them and then you download them at once. Okay, and then you click on download. Now I already have them, so I'm not going to click on download, but you click on download and then make sure that you find where they have been downloaded. And when you download them successfully, make sure that you put them in the bioinformatics folder. Now, mine are in the downloads here. When you open the downloads and you want to copy these files into your bioinformatics folder, pay attention to the RDS folder, which is a zip file. Now, when you're planning to copy them, open the zip file so that you can have all the other files that are in. So these are the files that you copy. You copy all of them and then you take them to the bioinformatics folder. Likewise, for the other zip file as well, there is this one, the sequences folder, this one here. When you open, you will find quite a number. You will find quite a number of other files inside here. These are the files that we want you to copy and you take to your bioinformatics folder. So you will copy these plus the rest of the files that you've downloaded. So when you go to your desktop, in your bioinformatics folder, you will be having what you've copied from the Google Drive and from within those other folders that are zipped. Make sure you open them, copy the contents from in there and place them in your bioinformatics folder. So your bioinformatics folder is going to have quite a number of items because we expect you to expand the zip files that are there, the 2 zip files, and copy the contents within and place them inside the bioinformatics folder, inside the bioinformatics folder. So I'm going to pause and see the link. Okay. Somebody is asking for the link. So the link to the Google Drive has not changed. We are using yesterday's link. We are using yesterday's link. We are using yesterday's link. So I'm pausing so that we can do the downloads. I'm pausing so that we can do the downloads.
Please, do you have WhatsApp link? Somebody's asking for it in Q&A.
Yes, I'm going to share the WhatsApp link. Okay, so if you are seeing, somebody's asking, can we go through the materials to download again? All right, so in the Google Drive, if you're seeing my screen, then you're looking at the Google Drive. Okay, so you can just okay, so we are downloading the silver FASTA file, the second silver file, the sequences file, the RDS folder, the metagenomics packages, if you haven't already downloaded those, the metagenomics activities, and the metadata. So we are downloading all these files here, these ones that are highlighted. Okay. Oops. Download all of them up to metadata. Somebody's saying the zip folder, when opened, still has other zip folders. Now, those zip folders that are inside that zip folder are the ones that we would like you to copy and put in your bioinformatics folder. Okay, there are many, and those are the ones that we will want you to put there. So when you download them, for example, I'm going to go to my downloads. When you have downloaded them together, they'll probably come in a drive. So the RDS folder is a zip folder. The sequences is a zip folder. So what I want you to copy is to open this, the sequences themselves, and you will find very many sequences here. Okay. I would like you to copy all of them. Control A, right click and copy, and go and paste them inside your bioinformatics folder. I've already pasted them. So your bioinformatics folder will contain all those files. You will find them here when you paste all of them, okay, including all the other files. So again, remember there are two zip folders here. There is one for RDS. So when you open the one for RDS, you will also find other files inside. I would also like you to copy all of them, highlight all of them, right click, copy, and you paste them in the bioinformatics folder. So mine have already been pasted. So they're, I think, below, they're down here. So your bioinformatics folder will have all the files. These zip files are coming from the sequence zip file. Make sure you expand them and copy the contents and place them in the bioinformatics folder. Okay, so now I can pause and see the progress. And see the progress. Okay, if somebody's zip folder has downloaded in the bioinformatics folder by default, I still would like you to open and copy because our working directory is going to be in the bioinformatics folder. So open them, copy them, and put them there. Somebody says theirs is taking long. Yes, I expect it to still be downloading. So please continue downloading. Continue downloading. But when you succeed in downloading, please type 1 there. When you succeed in downloading, please type 1. Okay, the link to the Google Drive. We are using the same link. We are using the same link to the Google Drive. Okay, so your bioinformatics folder in particular should have these items. There are quite many. They should have all these sequences, the FASTQ files, and the RDS files, including the other things that you have downloaded as well. Somebody is asking what files are we downloading. So as other people continue to download, the files that you're downloading are here. So the silver file, this one, the silver file, the sequences file, the RDS folder file, the metagenomics packages, the activities, and the metadata file. Some of the files are quite large, so we expect the downloads to be a bit slow. Just ensure that you have stable internet. But also let me know if you have succeeded in downloading the files. Somebody's asking which files. If you're looking at my screen, please look at my screen. The files that you're downloading are highlighted here. The files that are downloaded you're downloading are here, highlighted on the screen. We have the silver file, we have the sequences file, we have the RDS folder, we have the metagenomics, and up to the metadata here. All right, so I'll be checking through to see the progress. Now, this is important. You're going to keep downloading these files. So somebody says in the RDS folder, there are 2 folders inside this folder. Which one should we copy? Open the RDS, the one that is RDS folder, open it and copy the contents that are inside. Okay, so let me show you. So when you download the RDS folder, this one here, when you open it, there are 2. Yes, so it's the RDS folder itself that you're going to open. And when you're copying, you are copying all of them. Every RDS file that is in here needs to be copied, and taken to the bioinformatics folder. It's important that we have all the files that we're going to use during the sessions. Okay, so I'll be checking through the messages and try to understand. Okay, still downloading. I tried to extract the files from the sequence zip and selected, but they are seen like in folders. Okay. Yes, they are in zip folders. So I'm going to show you the sequence ones as well. So when you download, depending on where it has gone, you will find the sequences folder here. It is a zip file, but we want you to open this file. Okay, now when it opens, you open the file. So when you open the file, there'll be many other zip files inside there, and you copy all of them. Again, highlight all of them, Control A, right click and copy all of them. So you're going to copy all of them and take them to the bioinformatics folder. So your bioinformatics folder will have all the zip files within the sequences file, and there will be zip files. This is how they'll look like. And then it will have the RDS files that came from the RDS folder there.
There should be 24 files that are zip,
Yeah.
and they should unzip those ones because we'll use them in that.
Yeah, so just ensure that you bring all the zip files. There are many, copy all of them. Just highlight them all at once and copy them. Okay, progress with the downloads. Yes, if you've downloaded. Yes, if you've downloaded. 1 for yes. 1 for yes. Okay. Great. So I see quite a number that have downloaded and placed them in the bioinformatics folder. There are those that are still downloading. Okay. All right. So we are still giving time. In the meantime, even if we start the presentation, you will still have plenty of time to keep downloading. So just if you're downloading, just allow them to download. If you're downloading, just allow them to continue downloading. Allow them to continue downloading. It's important that they all get downloaded. Okay. All right. So we are going to continue to give us time to download. Now, allow this to continue downloading. I would like to know from those that attended the webinar yesterday, we had an activity that we were supposed to do. And in that activity, we were supposed to run the metagenomics packages file. In the comment section, did you manage to run them? Yes. 1 for yes, 0 for no. Did you manage to run the file successfully? Did you manage to run the file successfully? It will be important for us to run that file as well. Yes, no, no, no. Okay. So we have quite a number of people that did not run that. So I will open that file and show us what we need to do. And then I'll show us what we need to do. And then we will leave it running. Okay, so that's great. You cannot find the silver sequences and the RDS folders in your download. Did you download them? Have they succeeded? Do you have a notification that the download is complete? Because if the download is complete, then you should be able to find it. And meanwhile, if you download all of them at once, it will probably come as a drive download, a whole zip folder that contains everything that you've downloaded, which you need to double click in order to open up. So let me know if you have downloaded those as well. Okay. So if you're still downloading, that is okay. Allow the files to continue downloading. What I'm going to show you next, okay, is the assignment that you're supposed to have done yesterday. we all want to have that installed. So the metagenomics packages is the R file that we want to run. It contains the packages that we are going to use for the bioinformatics training. Okay, so with that, I will go to RStudio as you let your files to download. I'll go to RStudio. Now, for those of us who are new to RStudio, again, there are 4 windows in RStudio, and each of the windows performs different functions. This window here at the top will contain the scripts that we are going to use. This one below is where the code is executed. On the top, this side, we do have the environment, which will contain the different objects. Objects can be lists. It can also contain the data sets that we are going to use, okay? And then right below here, we have the files, we have the plots, we have different plots that will be here according to what we are doing. So make sure that you can see all the 4 windows. If you cannot see the 4 windows, display them using this pen here. If you cannot see the 4 windows, click on these 4 windows here, 1, 2, 3, 4. When you click there, click on show all the panes. You will find all the 4 windows. Somebody has that comment. Please let me know if you've seen them. You can adjust them accordingly. Okay, so now, so we are going to go through the assignment, and those who have installed the packages, we just want to see if you successfully run. Now, if you successfully run the file, the entire file, then you shouldn't be having any challenges. Somebody say, I'm not seeing the RStudio. Okay, I'm sharing my RStudio. Can you confirm if you can see, if the rest of you can see my RStudio? Yes, 1 for yes. Okay. Now, the person who is not seeing my RStudio, you may be having a challenge on your side. You may need to get in and out of the webinar if you are having challenges, especially with the internet. All right, thank you for confirming. Okay, so once again, I was showing us that this is RStudio, and for the environment, we will be seeing quite a number of items and data sets and data frames and lists and vectors that will be appearing here. Okay, so we expected you to run the metagenomics packages. Your metagenomics packages file, which is an R file, should be inside the bioinformatics folder by now. And you yourself, you should be working in the bioinformatics folder. You should be working in the bioinformatics project. That means up here, your RStudio should show the bioinformatics name, because we expect you to be opening that project. We expect you to open your bioinformatics R project. If you do not have a folder, then it means you didn't create that folder. And if you didn't create that folder, then you can create it now. It's a very fast process. Somebody says, how do I open my bioinformatics folder in R? You do not open your bioinformatics folder in R. You just open the project. So if you already have it and you created the project, then you go to the project, rather you go to the folder. I have so many of them open here, so I'll just try to close them. You'll go to the folder and open R from there. If you do not have and you've just opened RStudio, then you need to create the folder, okay? So for those that have the folder, we are going to open the file, this file, inside your bioinformatics folder on your desktop, there should be a metagenomics packages R file. This is what we want to open. This is what we want to open. You can just double click on it. Alternatively, not to confuse you, when you come to RStudio here, go to File, Open File, and search for it. It should take you to your bioinformatics folder. Then you can search for it here, metagenomics packages, then open it up. Okay, that's great. So if you have opened it and you hadn't done the installation, we would like you to run and install all the packages that are here. Okay, I already have mine, but in order to run all of them at once, Control A, highlight all of them, and click Run. Mine are done. So once the red button that is here,
Please,
yeah.
somebody type in the in this chat that how do they know if they have installed their packages successfully? So maybe you
Okay.
can also show them about the libraries. Thank you.
So they've installed the packages successfully if you do not have an error. We can load the libraries as well. We have the other file as well. First of all, before I go there, I would like to know if you have opened and run this installation. If you have opened and run this installation, yes, no. And if you're failing to run this, can you let us know if there is an issue? If you have an error, you can let us know as well. Okay, somebody says we repeat the procedure. So it's still running, okay. The procedure is still running. All right, so if it's still running, maybe we're going to give a few minutes for it to continue running. Please make sure that you are installing the metagenomics packages.r. Make sure you're installing the metagenomics packages.r. Somebody's asking from the sequence zip, what do you copy and paste? Open the sequence zip and copy and paste all the zips that are there. All the zips that are in there should be copied. All of them should be copied. Ensure that you are installing metagenomics packages. So if they're telling you it's not installed, click on install. If it shows that, click on install. Highlight everything here. Okay. Highlight everything and click on run. highlight everything and click on Run. Okay, so it's going to take time. R does not support to open zipped folders. Is that an error you're getting? Please copy the zip folders and place them into the folder that you have. How do I bring the metagenomics packages here? Okay, if you are bringing the metagenomics packages, let me first close this. Come to File. How do I install? My environment is blank. No, it's not okay. Your environment should not be blank. So if you are opening the metagenomics R file, come to File, Open File. Open File. And if you're working in the bioinformatics, you should find it. If you don't, you open the bioinformatics folder and look for it. Metagenomics packages here. Metagenomics packages. Click yes. All right, somebody's saying the red circle is stagnant. Which red circle? So the red circle that is usually down here shows you the progress that's being made in the execution of the code that you've run. So if it is still red, you need to leave it to run. It also depends on the internet. It also depends on the internet. The pop-up, click yes. Somebody wants to see the end of the script. Do you mean this? Okay, so this is the end of the script. If you wanted to see the end of the script. All right, so if yours is still running, you're going to have to leave it to continue running in the background if your internet is slow. By the time we get to use it, it will have finished doing the installation. By the time we get done, by the time we reach there to use those files, you will have finished installing these packages. You'll have finished installing these packages. So somebody is saying he ran all and it's still, yes, it is going to continue running until all of them are done. That is when the red button will go away. Does the order of where each box matter? I do not understand the box that you're talking about here. Maybe you can clarify. Okay, you mean the global environment? If your global environment is still empty, it means it's running. Now, if it's not successful, then it should bring an error down here, okay? If it's successful, you should have this statement here, all the packages installed and loaded successfully. These ones here, this statement should come if you have finished successfully installing and loading. If you don't see them up here, it means something didn't go right, and then there should be an error term. The red button will run depending on your internet, how fast your internet is, because it installs these packages. It requires internet. So all packages installed and loaded, yes. Now, when you have that message, then it means you are successful. If it is slow, allow it to continue. As we start the presentation, you will have time to continue with this. So I am going to stop here and I'm going to stop sharing my screen. I will be addressing some of those questions in the chat. Okay. Stephen, you can start the presentation.
Okay, thank you everyone, and thank you, Peace. Okay, let me share my screen here. Okay, if you can see my slide type 1. Okay, so yesterday we started bioinformatics. We defined what bioinformatics was, and then we watched a video about the central dogma, molecular biology, and that took us from how you start from the DNA. From the DNA, you come to RNA, then from NRA, you come to protein. So I'll give you an example of maize. So for a breeder where the maize failed and there was a drought, no rain, and few plants survive. So we wanted to find out why did those plants survive. So those plants survived because of the genetics. So the gene for drought tolerance, maybe they were activated. And then from there, in the process, the proteins were made, and those proteins were made, they were resistant to the heat. Then after that, we talked about the frames, how you translate the 6 frames of DNA to proteins. So we discussed the first frame, the second frame. So those are forward, first, second, third are forward, and the reverse for 4, 5, and 6. And we went online and did activity 1. And then after that, said, okay, after identifying your protein, what else do you do? So, okay, you can go to sequence analysis. One of them is by similarity search. So one of the methods that we used for similarity search was BLAST, basic alignment search tools. And we discussed different types of BLAST, and the most common ones are BLAST-N. That BLAST-N uses DNA. So you have your queries a DNA, and then you query the database also DNA. And then also for our activity, we use protein. So that is BLAST-P. So the query is the protein, amino acids, and then the database is also protein. And then after that, we say that, okay, sometimes the primary sequence, that's only amino acids is not enough. You can go to the next level. We talk about secondary structure. In the secondary structure, we talked about the 3 types. We have alpha helix, we have beta sheets, and we have coils. So that's where we stopped yesterday. So we're going to continue today with our sequence analysis. So the next is tertiary structure. Because we have our primary structure are the amino acids. Those are the 20 amino acids. And the secondary structures are those alpha helix, beta sheets, and coil. And tertiary structures now is at another level. So what's the tertiary structure? This is the 3D arrangement of all the atoms in the protein. So they're formed by folding secondary structures. So these secondary structures, they're folded together. They're alpha helices, beta sheets, and loop. So they're stabilized by hydrogen bond, hydrophobic interactions, ionic bonds, and disulfide bridges. So these secondary structures, they fall together. So in the end, the functional protein basically is determined by the 3D structure. Whether it is an enzyme or it is for other purposes, it's determined by the 3D structure. Okay. So for example, we talked about the drought tolerant maize. So for it to be drought tolerant, maybe it was determined by a certain type of enzyme that activated certain metabolism. So those were done by the 3D structure. Okay. So this is an example of 3D structure. You have these are the beta sheets, and then you have a coil in between, and then you have this alpha helices, and then something like that. So this is how the 3D structure look like in cartoon wise. Okay. Okay, why is this 3D structure very important? One, it explains the mechanism for enzymes. I've already talked about that, the receptors and transporters. So all the mechanisms of all those, either being an enzyme, being a receptor, being a transporter, is determined by the 3D structure of a protein. And then the active binding site, or the pockets of that protein, is determined by the 3D structure. The people who work with drugs in pharmaceutical company or who design vaccine, they always work with protein in a 3D structure. They don't work with protein in a primary structure, that's the amino acids, in the secondary structure, but they work with protein in the 3D structure. Also, 3D structure will help you to determine cause of disease, for example, in mutation. So if a protein is mutated, for example, yesterday when we were talking about translation, you have to have the 3D codons, okay, to determine what specific amino acid has to be added. So assume those 3 bases, one of them there was a mutation, maybe A is changed to something else. So instead of the genetic code, for example, if that specific amino acid was alanine, which is an A, because of that mutation, that alanine turned into something else, maybe another amino acid, maybe the glutamate. So remember, secondary structure is determined by also the type of amino acid. So if that is changed, the secondary structure will also change. And that secondary structure will also affect the tertiary structure of the 3D structure. So if that protein was responsible for not causing a certain disease, and because there was a change, that change will be identified from the 3D structure. Okay? And then another important part, people who do protein engineering or synthetic biology, they always work with the 3D structure protein. Okay. Another important function is identifying potential drug targets. So all the drugs that we take, most of all the drugs that you take in the body, most of their targets are really protein, by the way. So now if you're trying to develop your drug and you know the 3D structure of the target, will be easier to work with the drug so that they can bind with that protein. And also by medicine, sometimes people who have Alzheimer's or cancers. So those are sometimes are the result of modified proteins. Okay. Another good example in agriculture and biotechnology. You can engineer enzymes, you can engineer crops like BT resistance. All those are done using 3D structure. Another field that uses 3D structure is comparative genomics. So what do you mean by comparative genomics? So you can compare 3D structures of one species compared to these structures of other species. You compare their protein structure. So this is now possible. It used to be harder because as I mentioned yesterday, the hardest part of bioinformatics was predicting protein structure. Used to be done by experiment. But 2 or 3 years ago, Google came up with an algorithm that they even got a Nobel Prize. That now does a very, very good job in predicting protein structure. So now you can predict protein structures for all the protein in a species, and then you compare them. And then last but not least is for synthetic biology. Okay. So how do you predict protein structure? There are very, very methods of predicting protein structure. And a few years ago, we didn't have this tool called AlphaFold. But when AlphaFold came, it changed completely how people do biology now. So this AlphaFold is developed by a company in UK, but was bought by Google. It's called DeepMind. So what they did, they went to Protein Data Bank. Remember yesterday I told you about Protein Data Bank is where when you predict your protein structure experimentally, all the protein structure that have been predicted experimentally, the public one, because sometimes companies keep theirs, they're all deposited in Protein Data Bank. So using the algorithm, they trained millions of those, and then they were able to use the trained algorithm to predict this protein, and they're really, really, the accuracy is really, really high. So every year, people compete. So what happens is it's called cups for training. So when a protein is predicted experimentally, they give the sequences, the primary sequences to all these people who develop this algorithm to predict. And then they score them. So people used to do the computation, but the accuracy was not very high. But in 2014, in CAPS 14, that was the breakthrough. That's where this deep mind was able to do almost, we call it miracle in bioinformatics. For the first time, to have an algorithm that can predict a protein structure that is almost close to 90 something percent accurate. So this has been a very, very exciting in bioinformatics field, in drug discovery field, in other field that's related to protein function. Okay. So how do you predict protein structure using AlphaFold? They made it simple. You don't have to go through other steps because the way you use to predict protein structure, sometimes you have to get your protein, get other sequences, do the alignment, but they made it simple. What you only need to have is your protein in a FASTA format. When you have it in a FASTA format, you can either download their software, which is not advisable because it takes a lot of computational power, and you have to have at least a server to do that. But they were kind enough to work with EBI, and they have this online. So what you do, you go to the website, which I will show you. You get your sequence, and then you submit. And then after submission, it will run. You wait for minutes, it depends on your internet speed, or also the protein size, and sometimes also the queue. So it can take minutes to hours. Then after that, you get the output. The output will be the predicted 3D structure of your protein. And then you also get your confidence score, and that confidence score is called predicted local distance difference. So this is basically their algorithm predicting, giving you their confidence. So if you get a confidence of greater than 90 or greater, this is very, very high confidence. And between 70 and 90, this one is with good backbone accuracy, and less than 70 is lower, always loops or disordered. But if you can get something 90 and above, and if you, for example, I'll just go back again to maize. If you did your sequencing from those plants that survived the drought, you did the translation, you got your amino acids, and you did your secondary structure, and you submitted to DeepMind, and you got your score of 90 and above, then that basically will be good, because if you compare that to a structure of a protein that is been at least solved, that will give you with a very, very high confidence what that protein is. And from there, you can now start doing the downstream analysis why those maize plants survive, okay? So you can also download the results and use it with other software like Pymol, Chimera, and so forth, okay? So this is the online the tool of AlphaFold. Is very simple, and we will work with you together. It's very simple. When you go to the website, it just tells you AlphaFold protein structure database, tells home, about, and so every online software that you have, it will give you about the home, what it does, and the only thing you need to do is just paste your amino acid sequences here, and then you search, okay? After searching, of course, you see the progress going on. As I mentioned before, can take few minutes to hours. So you can just go start watching football, do something when it's running. And then this is what you get when it's done. So when it's done, it gives you basically a protein name, kind of a link. And then it gives you the score. This one's 83.69, which is high. And then also it gives you the query, the sequence that you sent, and then the sequence that's in the database, okay? So this is the first output that you see. But if you want to see the detail of the real results, you just click this one here, and then it takes you to something like this, okay? So here, you'll see a summary and mode of confidence. So it will see, these are the scores, but I would like you to pay attention to the predicted structure that's giving you. So when I submitted my protein, this is my predicted structure. So if you look at the blue part, that means the accuracy is very, very high. So when you put your cursor, it will tell you which part, and will also give you the what part of amino acid. So if you're maybe doing something like mutation analysis in the lab of this protein, it will guide you that the most accurate part that maybe you can do in your analysis would be the green part. So this blue part here would be the blue part. This blue part here, this structure here is very, very accurate. But you can see other parts here that are very low, like this part here, that one, if you're doing mutational experiment in the lab, you should not pay attention to this part here. And then some are light blue, you don't pay attention, but the most part that would be very interesting for your future work will be the part that are blue, because they have very, very high accuracy. So that is the summary. And then also, it can also, it tells you the similar proteins that are similar to what your protein are. So it gives you those so that if those proteins, somebody already done some work with them, so then you don't have to reinvent the wheel. For example, if the top protein here is similar to your protein, and somebody did a similar work, they published that protein, they have a paper, that paper, they have a methodology of doing mutational analysis that will help you a lot. So I think this is very rich because it gives you also similar protein and other things altogether. But I wanted to share this with you, how to interpret the results. Okay, so I'm going to go now and run the prediction with you. And then after that, we'll do activity together and then we'll move on, okay? So let me repeat that again. See if I have some question here. Okay, let me repeat that again. I haven't been to the website, but I'm going to repeat again. So this is the website for predicting 3D structure. And so far, the best algorithm in the world for predicting 3D structure. So they have, you can download and run on your computer, but you need a very, very fast computer, and it's not advisable and takes a big space to do that. So the easiest way, you can do this prediction online. So Google is collaborating with EBI in Europe. So anybody can use this. So the only thing you need, if you have your DNA, you translate it to protein because you need your protein structure, your primary protein in amino acid. And then you paste it in this window here. If you want to learn more about the AlphaFold, you can go to About and the rest. But very simple. You get your protein sequence and you paste it here. And then you submit. We'll do that later. And then when it's running, it's going to take from minutes to hours. Depends on your protein, depends on the protein size, depends on also your internet speed. And then when it's done, you get your results back. And this is how it will look. It will give you the top with the link with your prediction. And then it'll give you the protein name that's similar to what you have submitted. Also give you the gene and then the organism. And then also in the UniProt, it will give you the UniProt ID, and it give you the score. Remember, I told you the score of 90 and above is very high, and then 70 to 90. So this score is 83.69, which is high. And then on top will be your query. And then it's also like BLAST. It gives you the E value, the positive, and the gaps and so forth. And then to get into detailed, this one is a link. You click on this. If you click on that, it will bring you here. It's summary and model confidence. So that will give you the structure of your predicted protein. Okay? So this is the structure of predicted protein. I would like to pay attention to that. So if it's your predicted protein and you want to do more experiment in the lab, the part that you'll work with are the part that are shaded blue because they have very, very high confidence. So if you now want to do some mutational study for your protein, you'll work with some of these amino acids that's here. This one here is very low, so it's not advisable to work because the prediction is very low. This one, yeah, so this is to guide you. Okay. So you can click on, this is summary, and you can also see the domain. You can also see the annotation, and you can also see similar protein. Now that I will show you when we do the running. Okay. And then you can find similar protein. And this similar protein here, the top one is this one here. And then also give you the score, give the length. And if you click, it's a link, for example, this protein, if you click on it, it will take you to the source who submitted this. Sometimes it's already been published. So if you go to the paper and read the paper, so if they maybe did some mutational analysis, we'll use their method, okay? Okay, so I'm going to go there. So this is not the one. Let me go back. Just give me a minute here. Go back to our slide here. Should be activity 5. Let me go back. I'm gonna go back to my slide here and go to the website here. Let me go to activities. Activity 4. Okay, if you see my slide, you type 1. If you see the website, okay. So this is the website. So you can go to home, you can go to about, and so forth. So the only thing you need, I hope won't take long, is to paste your protein here. Okay, very simple. I'm going to pick one of the proteins here and use it. So I'm going to paste my protein. Let's take this. After pasting the protein, you just search. See this search here button? You search. So it's in progress. I hope it's not going to take hours. So let's just wait and see. Tree searching. Meanwhile, those who were installing the packages, can you check to make sure it's complete? It doesn't take long. So this depends on the internet speed, also depends on the queue, how many people are using the server. So you see that's going around, that means it's doing something. Okay, so meanwhile we're waiting for this, let's go do exercise here. So let's say at least we have activity 4. Yesterday you downloaded this file called activity, okay? So let's all do this together. I will when this is done, then I will explain, but let's do this together. You open your activity file that you downloaded yesterday, and if you click on this, it will take you to the website. And then just copy, don't copy where the session number starts from. Just copy only the sequence. and paste and run. So let's do that. So open your activity document that you downloaded yesterday. Go to activity 4 and click on this link here. If you click on that link, it will take you to the AlphaFold website. And then just copy from here and paste and search. So let's all do that together. So open your activity documents of yesterday. Go to activity 4, click on it, copy this sequence and run. So step 1, if you have succeeded doing that. Okay, good. Okay. So mine is still running. So let's make those run, and mine is still running. We'll give you another 5 minutes. That'll be our break. And then we can move to the next. We can move to multiple alignment. So I'll give you 5 minutes. That'll be our break, okay? 5 minutes break. Okay, mine is done now. Okay, so I would like you, after 5 minutes, I'll explain this, but mine is done now. Okay, so let's go back to my results. If you see my results, type 1. Okay, good. Okay, so this is the results. If you look here, this is the predicted protein called CC chemokine receptor type 8. And this is the protein name. It's a receptor protein. And this is the gene. Source of organism is Tupacia chinesis. Okay. And this is the UniProt ID. And this is the score, which is high. You know, score is high is from 70 to 90. And this is the sequence length. This is our query. So this is the query. You know, protein always start from M, but this is where it started aligning from. So our E value is less than 0.05. This is okay. So this is our alignment, 66% positive, and this is the gap. Okay. So the next, we want to go and see how our structure looks like. To do that, I'll click on this. Okay. So this now takes to my structure. So this is the structure here. You know, it's not a very good structure because if you look, there are very few blue part here, and I would like to watch this. So each time I move, you can see it gives me the position and the amino acid. I wish I could just, you see, you see the amino acid at position 106. Each time I move, it gives you where the amino acid is the name. So if I click here, this is very, very low. So this is very, very low and something like that. So that is my structure, but this is a very poor structure because the blue you can see here is not many. Light blue, okay, but most of this structure is basically alpha helix. You can see that. And now if I go up here, that is the summary of the model. If I click on domain, you can tell me which domain here. Domain, this is from this source. This is the domain from this Cathy catch. This is a database of protein and give the score. So if you would like to go further and see what type of domain it has, so it gives you that. So if you look at this domain here, so this domain, protein domain from that database it starts from this. If you click on this domain, it covers these amino acids from this position here where the red kind of pinkish, and that's the domain. Okay, so if you could look at that notation, it tells you that no annotation information available, and then you click on similar proteins. So this is the simple similar proteins with your protein, family A G protein couple receptor. So if I click on this, we'll take to what this protein is. And this is the structure of the protein. That's similar to ours. Okay. Let's go back again. So, okay, as any of you has succeeded, type 1, if your protein, if your structure is there, type 1. Oops. Okay, so you got your structure. Okay, thank you. So we're gonna continue. Okay, the last part of our sequence analysis is multiple sequence alignment. So we covered DNA, RNA, we covered protein, and the subunit of protein, that was amino acids. We did a sequence search using BLAST. We did secondary structure prediction. We did tertiary structure 3D prediction. And all along, we've been using only one protein. But sometimes using only one protein, you miss some information. So the next thing you can do is align your protein or DNA with other sequences, and that will give you more information, okay? So what's multiple sequence alignment? So multiple sequence alignment is aligning 3 or more biological sequences. It can be your DNA, it can be your RNA, it can be your protein. There's also a pairwise alignment. That's aligning 2 protein, or 2 DNA, or 2 RNA. But when it's a multiple sequence alignment, you have to list at least 3 or more. So why do we do that? We do that, one is to find conserved motif. Remember when we went for domain and we click on that domain, how did AlphaFold, how did it get that domain? Because the AlphaFold took our protein and aligned that protein in that database with all other protein, and that's how the domain came in, okay? So multiple alignment help in finding the conserved motifs, functional domains, and evolutionary relationship. So if you want to do phylogenetic structure analysis, structure prediction, or functional annotation, you have to do multiple alignment. You have to align your protein of interest with other protein. Okay, so beginning today and then tomorrow up to Friday, we're going to talk about metagenomics. We're going to talk about transcriptomic, RNA sex, and genomic. Alignment will come in. All those have to be aligned with the sequence in the database. We're starting with metagenomics. We're going to get our sequences from the sequencer. We're going to do all the analysis, and then to identify the species or the genuses or the phylum, we have to align them to the databases. When you're doing RNA-seq, we're going to get our reads from the sequencers. We're going to align those reads to our reference genomes. When we're going to study genomics, we're going to do GWAS. We're going to do diversity analysis. We're going to population structure. We have to do alignment to all our reads to the genome. So multiple alignment is very, very important. Okay? So if you line your sequences, this is example, just a cartoon. You're going to find sometimes those sequences that are aligned, all of them can have the same amino acids, or sometimes others have different amino acids with different colors. So when you're doing alignment, either DNA or proteins, the similar ones align together. The ones that are not similar do not align. And that's why sometimes when you do alignment, you see these gaps. Okay? So why do you do alignment? First of all, it reveals evolutionary conservation across species. Because human beings is different from chimpanzees because of certain genetic traits. And those are from genes. So when you align this human genomes or with chimpanzee genome, there are going to be certain regions of the genome or the DNA or the protein that will be different. Another thing is also identify critical residues. For like example, active sites, they tend to be conserved. Binding region, they tend to be conserved. So conservation means they have the same, maybe similar or the same amino acids in going together or the same DNA. It helps you to predict protein structure. It also provides insight in mutations. I mentioned that. I go back to the maze. So assume the breeder got those protein he was looking for. He sequenced the DNA. After sequencing the DNA, he translated the protein. So he got those ones will be the experimental, but the maize, those maize just died that could not take the heat. They're also going to be sequenced. So after sequence, you're going to have DNA from two groups, the ones that survived and the ones that didn't survive. So after sequence, you're going to align them. And then after aligning them, there's going to be a region where there's one region is going to have DNA from the ones that survive, it's going to be different. And then from the region that survive also going to be different. Okay? So that's why I say provide insight in mutation. Also, it's used for homology modeling. Okay. So another application is phylogenetics. So phylogenetics help us to see how we evolve or how organisms evolve. So it gives you evolutionary trees to help you with evolutionary history. So another application is functional genomics. So if you align your protein or your DNA, it helps you to assign gene or protein to function. Also, it helps the protein classification domain. So when we submitted our sequence to AlphaFold, our protein aligned to a certain domain. It did not align to any domain at random. So that one also alignment helps because the AlphaFold took our protein and aligned that protein in Kathy database with other proteins. Okay. So How do you apply that in agriculture? Crop improvement. So if you have the genes that are conserved, if you do alignment, they can be related to stress tolerance. I already talk about the maize. Disease resistance. It can also detect genetic diversity among crop varieties and so forth. Plant pathogen. If you compare pathogen genomes, you can find that the genomes of pathogens that are very aggressive, if you line them, their very virulence can be different from those ones that are not very virulent. So can also help you to track mutation. In animal health and breeding, the same thing. You can align those sequences, either DNA or protein, you can see the differences from the alignment and so forth. So alignment is very, very important in both plants, animal, and so forth. Okay, how do you do that? How do you align your sequences? The first thing you have to do, you collect your sequences. It can be your faster sequences or other sequences, but I'm going to talk about faster because that's what we're dealing with. And the good news is there are very, very, very many websites there that can do multiple alignment for you. But also you can download software on your computer. So you have to have more than at least 3, if you want to do multiple alignment, at least 3 sequences and so forth. And then you just upload to the server and you can configure. I mean, you can configure means you can play with the parameters, but I always advise to use the default. And you submit to your server. And then after that, it will come back and you see your results. Okay, so they say online tool we're going to use. But before we go there, I would like to discuss the output of the alignment always when it comes back. In most of the alignments, when it come back, you see these stars and column and dot. So always, if you see stars like this, that means fully conserved residue. What does that mean? That means when you do your alignment, all the residues will be the same. So that means they are conserved. So this sequence at this position has F. This one also has F, F, F at this position. And when you have a star. So F is phenylalanine. And then L is leucine, the same. So you have star here, okay? So when you look at this strongly conserved group, what does that mean? The strongly conserved group means the amino acids are either charged or hydrophobic. So if they're charged, they're aligned together, positive aligned together, negative aligned together, or they're hydrophobic, that means they hate water, or they're hydrophilic, that means they love water. So those ones, that means the star means they're the same, basically. And then the column, these 2 dots means they're strongly conserved group. They're either charged and so forth. And then you have the weak, that is very, very weakly conserved. And then sometimes if there's no alignment, then if there's no relationship among all the sequence, and then they put a dash, okay? Okay, so before the webinar, I went and looked at all these databases, but there's one that was very, very interesting for me. It's called Cluster Omega. It's a very interesting one. It's based in UK. So this one can do, your input can be protein, you can do DNA, you can do RNA. Some of them that I looked at, they could either do protein or can do DNA or RNA. So I chose this one. The only thing that you need to do is get your multiple alignment sequence in your first step format. You paste. Alternatively, you could just choose a file from your computer. And then I always tell people to leave these parameters just the way they are. Then you submit. And then after submission, you see this. So this website is the same website we use for translation. You see this running. Then after that, you get your alignment aligned. Like I mentioned, if there's nothing almost conserved or similar, you have these dots, yeah. It's dashes here, okay? So where there's 100% conservation, you have your stars, and where there's conserve, you have 2 and 1 and so forth. But it's the same thing that we did the other time. You can also show them in colors, okay? So I'm gonna go back again and talk about how you can submit this. So this is the link. You can submit either protein, DNA, and RNA. You can either paste or you choose your file on your computer and leave all these parameters the same. Then you submit. This is just an example where I did the pasting here. And then after that, the job will be running. Then you get your alignment results, okay? So you get your alignment results. You can see the alignment results. Each protein will give you different numbers. So the aligned part. So this one, the first two 29 amino acids, this one like 45 and so forth until the last one, okay? So this amino acids, the total length is 201. This protein total length is 201 amino acids. Is 201, this 217, and so forth. You can see them in colors, okay? So what I'm going to do, I'm going to run mine, and then we're going to do an activity. I'm going to go here, okay? So this is it's going to be protein. If I DNA, I would change to DNA. If I have RNA, I would change to RNA. But since this is my protein, that's what I'm going to submit. If I wanted to use a file, I would just click here and I go and get my file from here. But I don't want to do that. I'll cancel it. I'm going to paste my protein. So let me look for my protein here. So these are my protein sequences. This. I'm going to copy. I'm going to paste them here. I'm going to submit. Okay. Failure response. Bar request, please double check all inputs and submit again. Okay, something is not right here. So I think failure response. Server bad request, please double check your all inputs. So I think I'm okay. Let me see, maybe I don't have to leave a gap here. Let's do that and see if it's gonna work. Gap here. You know, when you always use Microsoft software and you paste thing from them, sometimes you get some trouble. But my sequences had gaps in between them. Let's see if the gaps was causing the problem. Let's try again, okay? Nope, something is not right here. Why is not working? This is correct. This is correct. I see my input supposed to be, the input supposed to be faster format, really. But it's not taking my sequence. Let me clear and start again. What I'm gonna do, Make sure. What I'm trying to do is I don't want to include a gap. And since I'm getting this from my Microsoft Word, hope I'm not introducing something there that is causing the problem. Okay, let me try again. Let me reboot this. Let me refresh. Okay, let me try. I don't know what's going on. Okay, so let me try with the US sequence that we're supposed to use. Let's see if that will work. Because mine has failed. But this was supposed to be your assignment. Let me try that. Because it ran. We try these sequences here. Okay, it worked. For some reason, my sequence from what I was using for training is not working, but yours is working. Okay, so these are the results here. So you can see the first few, maybe 20 or 30 sequences, they're all variable, except start from here. But I think let's go to the colored part to make it better. So if you look at this one is the first one that's completely conserved, the G here, and then some here, but you can see the difference here from the beginning of the sequence. Here, there are a lot of variation here. And then also towards the end, there are a lot of variation. So if you're trying to do some mutational study, you would get in the middle here data. Like this one is so conserved here. Look at this. This one should be functional domain. You can see this part here. It's so conserved. Okay, so something is going on for this protein here. Okay. Okay, so let's go and do some exercise. Let me repeat again, but we'll do exercise together. So let me go back my slide. So this is the website. If you click, we'll take you there. And then we are using protein. You paste all the sequences that you have in a FASTA format, and then you submit. After submission, it run, okay? After running, you get something like this. So the conserved amino acids for all this protein will tell you that, okay, this part here is important, either it's doing something, okay? So you can also look through color-wise because each color has a meaning, okay? So something like that. So we are going to do the last activity for sequence analysis together. So you open the file you downloaded yesterday called activities. This file here, the file called activity, this file here, go back here. this file called activity, the one you downloaded yesterday, it has this activity, the last activity has how many sequences? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So I have 13 sequences that you're supposed to align. So in order to align these sequences, you have to go and click on this website here. After clicking on it, come here and select all these sequences, okay? Copy them and paste and run. So let's do that. I'm going to give you 5 minutes, okay? Let's do that. So this is our last activity for sequence analysis. Let's do that, okay? Meanwhile, I'll also go and try to answer some question Q&A. Okay, some say this packages failed to load data to. So did the package say that it was installed? Because we haven't reached where you're supposed to load yet. The person will say this package failed to load. We haven't reached where we're supposed to load the package yet. As long as you have installed the packages, just leave it there for now. Okay? The same story here. So we'll get there when we are going to use. But as long as you have failed to, as long as you have installed the packages, right now don't try to load those libraries. Okay? Where are we copying the protein from? So the protein, you're copying them from the file you downloaded yesterday called activities. Yesterday, you downloaded a file called activities. And you did the activities 1 to 3 yesterday. Today, you did activity 4, that was predicting protein structure. Okay? So you open that file. So today you did activity 4, that was predicting protein structure using AlphaFold. And the last activity is activity 5. This activity 5, and these are the protein. Okay, these are the protein for activity 5 for multiple sequence alignment. So that was the question, where to get the protein from. Okay. Let me go look for more questions here. After the results of the protein structures obtained, how do I download those results, including structure and other summary statistics? Okay, so it depends on the server. Sometimes you give your email, then they send it to you. Sometimes you download the results. It depends on the server. That answer that. To remove the first line on each code. Can you try to remove the first line on each code? You can, if you remove the first line on each code, that means you are on each protein, that means you are removing the accession number, the identification of the protein. You cannot, because every protein, every FASTA format starts with this greater sign here. You need that to identify the protein. So what's the maximum number of sequences that can be submitted? It depends on the server. Some takes 100, some takes less. So that's why when you go to a server, at the website, you have to read what is their maximum. And sometimes when you submit and it's above the maximum, it's rejected. Okay? So help me with the link. So the link for all the activities are found in that file called activities. So every activity has a link on top. Okay, let me go back here and show you. So we have the last 2 days, we've covered 5 activities. So every activity has a link on top. So this was activity 1 for translation. So this is the link that will take you to the website where you do the translation. Okay? So activity 2, we did blast search. So this is the link to blast server. If you click on this, it will take you to blast. Okay? Activity 3 was secondary structure prediction. So this is the link to JPRED4. Okay? And this is the protein for the activity. Activity 4 was predicting protein structure using AlphaFold. This is the link, and this is the protein. And the last activity is activity 5 for building multiple alignment for this protein, and this is the link. So the activities, every activity has a link. Okay? That answer that. Let me go back again and see if I have any more questions. Yeah. How do you determine if the mutation is the multiple sequence alignment results? No, you cannot determine if the mutation is the result of multiple sequence alignment. The multiple sequence alignment will give you that, okay, this sequence at this position is different from the rest. So assume at position 10, every sequence of all those 13, every amino acid of all those 13 sequences D. It's D, 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 but one of them is a T. Then maybe that T is a mutation. But it will just tell you, but it's not the result of multiple sequence alignment. Multiple sequence alignment will tell you that at this position, this sequence is different. The amino acid is different from the other sequence. Okay? Okay, I think I'm done with that. So when you're done with activity 5 type 1, so then we can move on. Okay, so somebody say, please come again on an explanation of star. Okay, I'm going to go back and explain that. Okay? Explanation of star colon. Okay, let me explain that. Okay? Okay. Do you see my screen? So where there's star, that means you have the same amino acid in that position. That means that position is conserved for all the sequences. So where there's star, that means you have the same amino acid. And then where is a colon, 2 dots, that means they have similar property. If you look at if you look at my cursor here, let's start with this, where this is very conserved here. So this region is really conserved. Why? Because this F is found in all these sequences here. This L is found in all these sequences here. Okay, if you look at this, the same. But let's look at this dot here. So this dot is not as conserved or this, but they have the same property. So these are charged positive amino acid. These are charged positive amino acid. That's why they're similar. Okay? And these ones, they're not very similar to they're not very conserved to this because they have certain property, but they're not as close as this. And if there's nothing, that means just they're not conserved, and there's nothing much you can do about them. But these are the 3 stars, that means in that position, they have the same amino acids. Okay? That's the explanation. And then if there is none, any of the sequences is not there, then they put this dash, dash, dash to present the sequences that are missing in that position. Okay? Okay, so that brings us to the end of our first topic, bioinformatics and sequence analysis. So we're going to change the topic here. We're going to change gear here. We're going to start metagenomics. And I think that will take us to 5. We won't finish today, but we're going to start metagenomics, okay? Let me see if some people have some more questions before we move on. Someone still, please come again on gene position after alignment. Okay. Okay, so let me be clear here. I'm not talking about gene position. So this is a protein sequence. So where does a protein sequence come from? It comes from DNA. So where does DNA come from? DNA comes from a gene has been activated. So what I'm explaining here is I'm explaining the conservation of amino acid in this protein. It's not a gene, it's a protein. So this protein, how do you get protein? How do you get a protein? Gene has to be activated. When gene is activated, the DNA is translated. And then from DNA, we come to RNA. And then from RNA, the 3 codon will bring the amino acids to form a protein. So these are protein sequences. And they're represented by the amino acids. And then now, these protein sequences, they work because they have these amino acids that are conserved and they form the 3D structure. But now if you get your protein that you don't know, you align them to the protein that maybe have been known. So now when you look at this, you can say, okay, this part here is the amino acids are also conserved. That means something's going on. These are conserved here. Something's going on, these stars here. So this should be maybe a functional domain for this protein here. This can be a functional domain and this can be a functional, but here there's nothing here. There's so variation here, that means they might not be doing anything. So I wanted to clarify here that what we are seeing here is not gene conservation. We are seeing amino acid conservation in these proteins, okay? I think that's I had to clarify that from the question. Okay, so let me stop here and we move on to another topic. So that was sequence analysis. So everything we'll do, we'll build on this, okay? So our next topic is metagenomics, okay? So if you see my slide, type 1 in the chat. Thank you. Okay, so I call this microbial community data analysis, and we call it metagenomics. So this week we're going to cover metagenomics, transcriptomic, which is RNA-seq. It will conclude with genomics. Okay? So what's metagenomics anyway? So metagenomics is a study of genetic materials recovered directly from environmental samples. I want you to think a minute about environmental samples. So when we talk about environment, we mean that where you're going to get your sample, it can be from the gut, it can be from soil, it can be from forest, it can be from the skin. It can be anywhere that you're going to get that sample. So you're going to study genetic materials from that. So why do we do that? In metagenomics, you're studying microorganisms, by the way. Here's the problem. The vast majority of microbes cannot be cultured. What do I mean by cultured? So culturing, you get your sample, you put them on a plate, you grow them, and then you plate them. And then after that, you get and then you sequence. But majority of microbes cannot be cultured by plating because of very, very, very many factors. So the traditional method before sequencing came, if you want to find out if this microbe is around, what you do, you collect your sample, you put them on Petri dishes, plating, you put them in Petri dishes, and you let them grow. After growing, you get those samples and then you send them for sequencing. But that's the limitation of the traditional methods. But metagenomics came, now you don't have to go through all that. You bypass the step of culturing. You don't have to culture. You get your sample, you extract your nucleotides, you just send them for sequencing. So that has been a very, very big leap with working with my microbes. So the study of genetic materials recovered direct from environment samples, no culturing. Okay. So this is how metagenomics in a nutshell is done. There are two types of metagenomics, which I will explain later. So one is called amplicon sequencing. And that one uses what's called markers. We'll discuss those markers later. You get your samples, use markers, and then after that, that's where the bioinformatics come in. You can use your chime, mother, and the rest. And then there's one is called shotgun sequencing. You get your samples, you just shotgun, that means you just crush them. And then you do your bioinformatics. You do, after that, you do your statistical analysis. So there are two types of how you do metagenomics. We're going to see them later, okay? But before we go further, I would like to talk about some definition. Because sometimes people use them interchangeably, but sometimes they use them wrongly. So what's the metagenome? The metagenome is the genetic information of a population that constitutes the whole genome of many organisms. So if you take a sample, maybe from a forest, the metagenome is the genomes of all those organisms in that environment. And the microbiome is the totality of the microbes that the genetic information has and how they interact. That's the microbiome. And then the microbiota is the collection of microbial organisms from a defined environment. So microbiota can be from forest, can be from the gut, and so forth. So sometimes people use this interchangeably, but they are not. So the metagenome, the genetic information of a population, that constitutes the genomes of many organisms. Then the microbiome, the totality of the microbes, the genetic information on how they interact. In the microbiota, the collection of all these microorganisms from a defined environment. Okay? So, metagenomic approaches. I give you briefly how the two types. The targeted sequencing, that's the most commonly used because it's cheaper, and the shotgun. So how do you do targeted sequencing? So those are done using markers. And the three common markers, 16S RNA, 18S RNA, and interspecies ITS region. So in order to work with those, we have to use PCR primers. So you sequence those, one of those markers, use those primers to amplify them. So they're successful because those markers, they have conserved regions and variable regions. So those regions allow you to identify a group of organisms. But when you do targeted sequencing, you have challenges because it's very, very difficult to identify species level because you're just dealing with only one part of it. Okay? So that's the disadvantage of using 16S or 18S RNA or ITS. But it's very, very efficient, very cheap, and most metagenomic is done this way. And that's for this webinar, we're going to analyze data from 16 RNA, 16S RNA. So the targeted sample is called amplicon sequencing because you use amplicons. So you use PCR, you create these amplicons, and then they have to be multiplexed. Multiplex means they have to be indexed or pooled. Why do we do that? Because sequencing is very expensive. So instead of just sequencing from one or few labs, they sequence from very, very many labs. So how do you identify them? You have to multiplex them. You have to index them. So when we go to real analysis, there's somewhere that I will tell you that they have to be demultiplex. That means you have to identify them by what? By their index. And this is done by barcoding. So if somebody talks about multiplexing in metagenomics, especially when they're doing amplicon sequencing, that means they're just indexing them. Because when they're sequencing, it will be very, very expensive to sequence just lab by lab. Okay? So before multiplex amplicon must be transformed into libraries, so you add your adapters, and then you enrich your region via PCR amplification. Okay. Okay. So without these markers, you will not be able to do your amplicon or targeted 16S. It's a targeted metagenomics. So I'm going to take some time to talk about these markers here. Okay. So let's start 16S. It's most commonly used when you're doing targeted metagenomics. So this 16S is part of small subunit of ribosome. It's found in prokaryotes, mainly in bacteria and archaea. So it contains both highly conserved region and hypervariable region. That's why that's why it's used as marker. So the presence of these high conserved regions allow researchers to design primers. That's primers that's specifically for that part. And then the hypervariable region is for that choice. So that's why when people are doing metagenomics in microbes, especially in bacteria, they use 16S because of those two regions. So the hypervariable regions now allow the researchers to to detect different species in the sample. Okay? So this is how you do it. So this is 16S. So it's very large, 1,500 base pairs here. So the conserved regions are kind of like this greenish. And then you have V1 up to V9. These are variable region. So what people do, you can just pick one of these. So the data that you downloaded, the sequence zip data, that piece made you download came from V4 region. Okay, so you can use any of these. So the primers came from this conserved region, but those sequences came from V4. So the 16S RNA is used as markers because of this. It's a very big 1,500 base pairs and has these variable regions here that you can use for your metagenomic studies. So the data set that we're going to use came from V4. Okay. 18S. So 16S is for prokaryotes, mainly bacteria and archaea. 18S, this one from eukaryotes. So it's subunit of also of ribosome. So it's homolog to 16S, but it contains the conserved region V1 to V9 except V6. If you look at the 16S, it has V6 here, but 18S doesn't have that. Okay. So when you're doing metagenomics, mainly in eukaryotes and fungi, you use 18S. Okay. So it's a gold standard for when you're doing metagenomics in eukaryotes, molecular phylogeny. And then 16S is for bacteria. That's what we're going to, our data for 16S. So this is the flaw of when you're using 18S. So you isolate your DNA, you do a PCR amplification, you do the purification, the product, library construction, then you send for sequencing. It can be nanopower, PacBio, Illumina, and so forth. And then bioinformatics picks up from here. So all this part is lab. This is sequencer, can be any company, and bioinformatics will pick up from here. And now you do your composition, community analysis, diversity analysis, evolutionary and functional analysis. The last marker is internal transcript spacer. It's called ITS. So this is located in RNA gene transcription region. So it's basically difference in base pair among genera, species, and strains. So sometimes when we want to get difference in genera, species, or strain, use ITS. So it's very easy and small to analyze. So it has been used mainly for different species in fungal genera. So people who work with fungi mainly use ITS markers. A lot of them use ITS markers. So this is the workflow when you're using ITS markers. You get your DNA, design and synthesize your primer, PCR amplification, library preparation, it is sequencing, and then the bioinformatics take over from here. Okay, so briefly, that was amplicon or targeted sequencing. Next is shotgun. It's called shotgun because you just throw things at it. It's just like using a gun. So this non-discriminate is sequence everything in the sample. So this is the good thing with this one. You can assign taxonomy, you can quantify organism up to species level. Remember, when I was discussing targeted or amplicon, you use on a region, you miss, but this takes everything. The problem is very, very expensive. It's very, very expensive unless you have a lot of money to do this. It's very, very expensive to do shotgun metagenomics. So 80% of the people who do metagenomics, they do amplicon or targeted sequencing. This would be perfect, but it's very, very expensive. Okay? So why are we studying metagenomics anyway? So metagenomics has applications too. So in agriculture, how can I apply it? It can enhance our research for disease resistance. You can find out what microbes are there, improve soil health, sustainable farming, guide use of biofertilizer and biopesticides. So the data sets that we are going to work on, I got from my colleague. She worked with biopesticides, by the way. So the data sets that you downloaded was for one of my colleagues from Ohio State. It's from biopesticide. She works with biopesticide. Okay, you can also use for human and animal health. You can categorize microbes in the gut, in skin, oral, or respiratory. You can also, people now using metagenomics to find microbes that are responsible for obesity, diabetes, and cancer, and so forth. Even in antimicrobial resistance, metagenomics is helping a lot to identify resistant genes across human and animal zoonotic, One Health surveillance, and so forth. So it has become part of research. That's why we study them. In environmental science, you can use it to reveal soil fertility, nutrient cycling, carbon sequestration, and so forth. Even in biotechnology innovation, you can discover novel enzyme for industry, new antibiotics, pharmaceutical, support genetic biology, and bioengineering. That's why we study metagenomics. It's applied in all these fields. Okay? Okay, so that was briefly about metagenomics. Just introduce metagenomics, but we are bioinformaticists here. So how do you analyze the data? Because I don't work in the lab. So as a bioinformaticist, it would be nice to work with people in the lab, see how they, of course, design that experiment. But at the end of the day, somebody's going to, that data has to be analyzed. That's why now have the data that has been collected has to be analyzed. So in order to analyze this data, we need to have databases. We have to need databases. So if somebody in the lab want to identify the samples, what type of organism or genera or species that's present in the samples, they should have at least a reference to compare with their samples. Because after doing the sample, getting all your sequences, you have to compare it with some databases. So there are three common databases that I use in metagenomics. The one that you guys downloaded is called Silver. So Silver is used. The Green Genes is one of the databases that is used, and the one is Unite. But Silver is very, very common. So when people told you to download databases, I think two of them were from Silver. So when you get your sample, you extract your DNA, send for sampling. After doing all this bioinformatic analysis, you have to compare your sample with the samples in the databases. And we have people who have been working on that. So these are the common databases that are using metagenomics. Okay. So what are the file formats that are used in metagenomics? Metagenomics is a field where when in bioinformatics sometimes becomes a little complicated because it has 8 different type of data that they send it to you. When you're working in the lab, some labs can send you a FASTQ file. These are raw reads. So we are going to work with FASTQ file. Remember when Peace told you to download that sequence zip file and you unzipped and you copied those 24 files, those are FASTQ files. But sometimes when you're working with somebody somewhere, they're going to send you a FAST file. It can be context, assembly, and so forth. And sometimes they do part of the analysis, they send you a sum of BAM alignments. That's why we covered alignment, by the way. They send you an alignment file. Sometimes they go to the extent of analyzing the data up to the variant call file, and then they send you this. And sometimes they send you this type of file. So it's hard to work with in metagenomics because you have to be able to work with all these type of files. It depends on who is going to send you the data. But always I would prefer somebody send me this FASTQ file because this comes direct from the machine. Because others from this one down, that means somebody already did some work. Okay, so for us, we're going to start for this metagenomics here in this webinar. We are starting with FASTQ file. That means the file came from right from the machine. And then sometimes, especially in developing countries, people, when you send your sample, they always send you back files either in Excel or in a comma separated value. After almost doing everything, they send you sometimes or OTU, which we're going to see later. So I want to share with you this type of files, but we're going to work with FASTQ files. Okay, so operational taxonomic unit, OTU. So this groups similar sequences together in metagenomic analysis. So most of the time when we send our sequences out, especially from developing countries, this is what we always get back after they did almost all the analysis. So what does it represent? It represents a class of DNA sequences, either from 16S, 18S, or ITS genes that are considered coming from the same species or taxonomic unit. And they're basically 90%, 97% common. So they always send you them in a form of a table, either in Excel table or in a comma separated value table. So all the other processes that we're going to go through, you don't see them. And most of the time, that's what they send you. Okay? So this is how the OTU table looks like. So you have your rows and your column. Your rows represent the cluster of maybe of a bacteria. So the first cluster is going to represent E. coli that have been found in your sample, and that sample contains 97% of that cluster is E. coli. And if you have 3 samples, we have 500. If you have your sample, the units here, the number of OTU, the volume number of abundance, maybe it can be the sequence lengths or DNAs. So they'll give you all those information. So sometimes they'll give you that units, the number of reads that are assigned to that OTUs. So for this E. coli cluster, the number of reads in sample 1 were 500, number of reads in sample 2 was 600, number of reads in sample 3 was 550. And then they go to the next cluster. In these clusters, 97% of these were bacterioids. The number of reads were 300 and so forth. So they send you this. But sometimes they send you what is called relative abundance. Okay, so what's relative abundance? Sometimes we hear about relative abundance. So relative abundance is basically the percentage of OTU counts per total reads. Okay, so I want us to go and be very simple here. So assume we sent our sample. We have 3 samples. We collect our samples from the forest. We have 3 samples. We got our DNA from those samples, send them for sequencing somewhere. And then what they did, they wanted to present our results in OTU. And what does OTU means? Operational taxonomical unit. So it represents your DNA sequences that I got from your markers. So they assume that they're 90%, 95% similar. Okay. So in our OTU1, 97% of that cluster is E. coli. In that sample 1, we had 500 reads because you send 3 samples. Sample 2 had 600 reads, and sample 3 we had 550. And then in OTU2, the cluster was bacterioids. So this is a different bacteria from the first one. So the reads were 300 and so forth in sample 3. But using these total numbers is biased. So what they do, they convert them to relative abundance. So relative abundance is just you take the total of everything and now convert them in percentage. But how do you do that? So in sample 1, the total of E. coli was 500 reads, bacterioids 300 read, and lactobacillus 100. So our total is 900. In sample 2, the total is this. In sample 3, the total is. So to be fair, we have to do the relative abundance. So how do you do that? You calculate the percentage of it. So E. coli in this column will be 500 times 900 times 100 will be 55.6%. And then bacterials be 33.3% and so forth. And in sample 2, this is percent of E. coli in sample. So now, each time you read a metagenomic paper, they don't talk about their total, they talk about relative abundance. It's always represented in a graph like this. So if you look at sample 1, the relative abundance of E. coli is around 50-something% here. And OTU for bacteria is around 33%. And lactobacillus around, let me go back here. E. coli 55.6%, bacteria 33.3%, and lactobacillus 11.1%. So when you read metagenomic papers, that's how it's presented. So in our sample 1, E. coli is 56%, bacteria is 33.3%, and lactobacillus is 11%. So this is now the relative abundance in this sample that we collected, okay, after doing metagenomics. Now we go to sample 2. The total of sample 2 was 1,050. So the relative abundance for E. coli is 600 divided by 1,050 times 100. Bacteria 400 divided by the total times 100. Lactobacillus is the same. In sample C, the same. So when people talk about relative abundance, which we're going to talk about we're going to see that tomorrow. We'll calculate relative abundance for genus and phylums. That's what we are doing. So for sample 3, bacteria is a little bit higher and so forth. So that's what relative abundance is. So when they're reporting OTUs, they report them in relative abundance. Okay? Okay. Diversity analysis. So when you do microbial study, you only have these questions here. How many species or OTUs or SVs are present in my samples? What's the level of phylogenetic diversity within each sample? How many similar or different samples or group of the samples from one another? Which factors, example, pH, elevation, blood pressure, and so forth, is affecting the composition of different microorganisms in the diversities? So all these questions that you ask are typically addressed in what is called diversity. And there are two types of diversity in metagenomics. One is called alpha diversity, and one is called beta diversity. So what's diversity? Let me go back. What's diversity in metagenomic? We have already seen relative abundance. That's one of the analysis that they do in metagenomics. We calculated that. Next is diversity. So when you are studying in metagenomics, you want to ask these questions. How many species, OTUs, we are going to talk about ASV later, present in my samples? What's the level of phylogenetic diversity within each sample? How similar or different are samples or group of samples from one another? Remember, when we were talking about relative abundance, we had 3 samples. We went to the forest, collected those 3 samples. You came to the lab, extracted DNA, send those samples for sequencing. They send them back after sampling. You did analysis. You counted the number of reads of sequences in each sample. And your OTU of 97%, you found that you have E. coli you have bacteria, and you have lactobacillus. And then you converted those into relative abundance. So you ask those questions. And then how similar are these samples? What factors? Because those samples are different because of different factors. So to do that, you have to address them with what we call diversity. And there are two types. There's alpha diversity and beta diversity. And we're going to look at each of them. Okay? Alpha diversity. Alpha diversity describes the diversity within a single sample. So we had 3 samples from the forest. So what alpha diversity will do, you go pick sample number 1, and within that sample, it will find diversity within that sample. You forget sample 2, forget sample 3. And then go and do the same for sample 2, the same for sample 3. So it's this describe diversity within a sample. So how do you describe the diversity? One is richness. What does it mean? The total number of distinct species present in environment. And it's always done by a metric called Chow index. That's within that sample. Forget about sample 2 and sample 3. The next is evenness. The uniformity of species abundant within a sample. And the common metric is Shannon index. And the third is dominance, or even account for both richness. So that one accounts for both richness and relative abundance. So if somebody's talking about alpha diversity, they're talking about diversity within that sample. So I have 3 samples. The alpha diversity is going to be for alpha diversity within sample 1, within sample 2, and within sample 3. So I'm going to explain it using this graph. So let's take sample 1. Sample 1 has how many? So the colors are different types of bacteria. Let's put it that way. So this one have high richness. High richness means how many bacteria? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It has 10 different types of bacteria. It's very rich, but it has low evenness. What does that mean? That means this blue bacteria takes a big chunk of all this. The number of reads of this bacteria is higher than the rest. Its richness, that means it's it has very many bacteria in these samples. But the evenness is low because this blue bacteria takes a big chunk of it. Let's go to this one. This one have high evenness. So we have all these bacteria still here, but you can see the distribution all of the same. This one has high evenness. And then dominance, it has very low richness. How many bacteria? 1, 2, 3. And it's only dominated by only 1. So that's what alpha diversity means. So within this sample 1, if it is very high richness, that means it has very bacteria, but also it's low, the evenness is low. So it has many bacteria, but there's one that's dominating. Okay? And this is calculated by Chow index. And then if within sample you have very high evenness, that means you have very many bacteria, but their distribution are the same. So this will have maybe 300 reads, 300 reads, 300 reads, like that. But if this sample has only 4, 3, 1 is so dominant, so it has very low richness, but there's also 1 dominance here. Okay? So high richness, low, so many species are present, but 1 species dominate. High evenness means species are present in roughly equal proportion. And dominance, only few species are present and 1 is overwhelmingly abundant. So that's alpha diversity within a sample. Okay? So beta diversity. So beta diversity now measured diversity, the similarity between the samples. Now we're measuring differences between sample 1 and sample 2, sample 1 and sample 3, sample 2 and sample 3. So now it helps us understand how microbial communities differ across environment, individuals, or condition. So that's what beta diversity does. So the common measure brackets the similarity, and it's based on species abundance. Okay? So the difference between alpha diversity and beta diversity, alpha within the sample and beta across the samples. Okay, so this is the difference. So we have like an example here. This sample 1, okay, we call it sample A, sample B. Let's just put it that way. So this is let's put this is bacteria 1, taxon 1, taxon 2, taxon 3, taxon 4, taxon 5. So in sample A, the abundance of this one compared to sample B, this is it's around 30%, this is like 10%. You go taxon 2, this is 10, this is more. So you're comparing between samples. Okay? Okay. So how do you work with the beta diversity? So we always do that through visualization, sometimes coordination. So the most commonly applied methods include principal coordinate analysis, sometimes called multidimensional scaling or MDS. Another one is called non-parametric multidimensional scaling or NDS, and principal component analysis. So when you're comparing different samples, you use most of these methods here. Okay? So this is an example of principal coordinate analysis. So you use distance. So for example, you're plotting distance group A and group B. So group A, you're plotting the number of reads here, and group B, so you can see that they're kind of separate. Okay? The principal component analysis, the difference between principal component analysis and principal coordinate analysis is principal component analysis uses variances, and principal coordinate analysis uses what? Uses distances. But both of them are used for beta diversity. Okay? Okay. So sometimes you have to go beyond that, the visualization. So there are other methods that I think we have covered them when we're doing data science that are used for analyzing differences among samples, such as beta diversity. We have univariate statistics. You have t-test. You have Wilcox test. You have Kruskal-Wallis test. Basically, what we covered when we are doing in data science, going beyond visualization. Or we have you can do box plot. You can also do Manova, which is equivalent to Manova. So all those are the statistical tests for beta diversity. For example, on the lower right here, you're doing the box plot for group A and the box plot for group B. And then on this side, you're using the principal coordinate analysis, and also you're using the Manova, using the p-value. So what we discussed in data science, you can also apply it in metagenomics when you're working with beta diversity. Okay. So other tests that you can do dispersion, which is test for variance. You can do distance matrix. You can also do ANOVA. You can do permutation-based analysis of variance, and so forth. You can calculate R statistics. Okay, so I'm going to stop here for today. And then tomorrow we'll pick up, that's where we're going to start using analyzing all this. But I'm going to spend the next 10 minutes or 9 minutes answering questions. So for today, I'll stop here. Then we'll pick from here tomorrow. Tomorrow we'll be using RStudio. Okay? So from now, I'm going to go and start answering some questions. Let me go there here. Okay. I need to answer some few questions here. Somebody say, please share the materials for metagenomics. We'll share that tonight. Let's see here. I'm just trying to see if I can get some questions here. So
in the chat. In the chat, there's a question.
In the chat, after the result for protein structure is obtained in alpha, how do you download these results, including structure and summary statistics? So that one, I'm going to go through and answer that tomorrow. Okay. So that one, I'll bring that tomorrow. I have to download it from from AlphaFold. Somebody's asking about. Let's see. Okay. Somebody's putting that. So what if I want both bacteria and fungi? So for bacteria, use 16S. For fungi, use ITS. Okay, bacteria 16S, fungi use ITS. Okay. I have more questions here. Let me go here. Okay. Okay. So please throw more light difference between PCA and PCOA and PCA. Okay. So let me talk about that. Okay, PCA and PCOA. I'm going to talk about that. Okay. So with principal coordinate analysis, you have to get the distance first. For example, let's take this sample, group A and group B. So you're going to use the Bray-Curtis methods that calculate the distance between group A and group B. So after calculating the distance between group A and group B, the result of the distance is now going to be used to draw the principal coordinate analysis. Okay? So one of them use distance metrics because the distance metrics will come like this. You're comparing the distance between all these samples within the group. So that the distance matrix is now the one that's going to be used for calculating principal coordinate analysis. But the principal component analysis will use the difference, the variances in the sequence themselves, not the distance, to calculate the coordinate. Okay? So that's the difference. One coordinate uses the distance, and principal component uses the what? The variance between the groups. I hope that answers your question. Okay? So let me go back and see if I have more questions here. What do you do with the gaps after alignment? There's nothing much you can do with the gaps. Sometimes you can align them manually, but it's not advisable. It's not advisable. Does AlphaFold make protein structure prediction methods such as no, it does not. It does not. Those experimental determination of protein, we still need them. With that dose, okay, let's be honest here. With that PDB, with that protein data bank, there would not be AlphaFold. So we should thank PDB for doing that. So we still need the protein to be, we still need those experimental. So they will not be redundant, by the way, they won't. So I think that answered that. We still need them. Someone say, the normal said, my problem is interpretation with the output. Well, I can see letters which I don't know what they mean. So those letters are amino acids letters. And each letter represents an amino acid. And amino acids, the proteins are made of amino acids. If you were here yesterday and you watched that video, how you come from a gene is activated in the nucleus, and then transcription takes place. From transcription, you get your messenger RNA and it translated into translated into protein. So those are not just letters; they represent amino acids. Okay. I hope that answers your question, Anonymous. Okay, so someone say, please come again on gene position in alignment. The alignment that we had is not about, we didn't align genes. We aligned protein sequences. And those protein sequences are represented by the amino acids. So where you see stars, that means they have the same amino acids among all those sequences. So we did not align gene, we aligned protein sequences, okay? Once we have results, how do we supposed to interpret results so that we can understand it, please? So when you have your results, where you have those stars, that means those are conserved area. They can be area that are very important for that protein. They can be enzyme, active site. They can be domain that is doing something for that protein. Please, the package failed to load. So we are not loading those packages yet. We will load them tomorrow. How can we extract DNA without culturing in case of fungi? You don't need to culture. You extract your DNA. There's a process where you extract your DNA without culturing. Share the slides. Okay, we're going to share the slides. I think we have run out of time. I can now get you back to Dr. Rukwara to take over from here. Thank you.
Yeah, yes, there are so many questions which are coming in. I think maybe what I can advise is people, they can have their questions tomorrow and then put them in the question and answer so that you can be able to follow up on them and respond to the people. So, yeah, so I would like to thank all of you people who have managed to attend this webinar, and I am seeing that you are appreciating this, and would like to thank Dr. Alpio and Peace for this training. Let us meet tomorrow again at the same time.